Welcome back. Today we're going to be talking about cross-bridge cycling and energy for muscle contraction. So in the last video, which was called, I don't know, something about actin and myosin, or myosin loves actin, something like that, we learned about how calcium is super important in regulating whether muscle contraction happens or not. In the absence of calcium in the sarcoplasm, tropomyosin covers the myosin binding sites on actin, and troponin is attached to tropomyosin. And so when this situation is happening, when there's no calcium in the sarcoplasm, no muscle contraction happens. However, when calcium was released, and if you remember, calcium gets released from the sarcoplasmic reticulum, calcium is going to bind to troponin. So when calcium binds to troponin, it actually changes troponin's shape. That binding of this ion causes troponin to change shape and kind of pulls it away. And in pulling it away, it also pulls tropomyosin off of these little myosin binding sites on actin. So once that happens, myosin is free to bind to actin. And so we go through this thing called cross-bridge cycling. The cross-bridge is formed between actin and myosin, and it's how myosin is able to pull on actin and shorten the sarcomere. So your book has it a little bit incorrect, but I'm still going to use this graphic just because it's something that you can see in your book. So we're going to actually start here in this area. So this, even though it's number 2, we're going to start here. So as soon as tropomyosin gets pulled away, the myosin head is going to reach up and it's going to grab onto actin. Now, the myosin head has been sort of cocked, meaning if you imagine your hand, if you wanted to pull on something, like pull on a phone cord or something like that, you would reach your hand out in front of you and then pull whatever it is back towards you. The myosin head is sitting there with its hand already out by the time it binds to actin. So as it does that, it's going to bind and it's going to pull actin in that direction. Very similar to the way that you would pull on a rope if you were playing tug of war. In the next step here, what's going to happen is that as the pull happens, ADP gets released. So ADP is bound onto each one of these myosin heads, and as myosin pulls with one of the heads, it's going to release ADP. And that leaves room for ATP to come in. Now remember, ATP is high energy. That's why we have this fancy energy, like exciting looking star around it. When ATP binds to that myosin head, it's going to cause it to release actin. So this head right here will release those myosin binding sites and release actin. Remember, ATP has a lot of stored energy in it, and when we break apart ATP into ADP, that energy that gets released, remember we have energy, that energy that gets released is used to recock the head. So imagine when we first grabbed actin, the head was cocked, meaning your hand was out. Then you pulled it towards you, you released actin, and then you reached back out. You bound ATP, hydrolyzed it to release the energy, and used that energy to reach back out and grab actin again. So in this way, you can almost imagine each one of these heads is like your hand, where you're hand over hand over hand, grabbing at actin and pulling it. And this goes on as long as calcium is in the sarcoplasm. At some point, calcium is not going to be in the sarcoplasm anymore. So we're always pumping calcium back into the sarcoplasmic reticulum. As long as we have the signal to do muscle contraction, even though we're pumping it into the sarcoplasmic reticulum, those gates are open and it's coming back out. It's almost like trying to pump water out of a leaky boat. No matter how much you pump out, you're going to have water coming back in. Same thing with the SR. We're going to pump calcium back into the SR and then some of it's going to leak back out. But as soon as you get rid of calcium, all of this changes. Once calcium is gone, troponin is going to change shape. It's going to allow tropomyosin to slide back over and cover these myosin binding sites on actin, and they won't be available anymore. So what happens in cross-bridge cycling is you can release the actin, you can bind to ATP, you can hydrolyze the ATP to allow you to recock the head or reach your hand back out again. But when you go to grab actin again and pull again, those binding sites are now covered. There's no place for myosin to bind on actin. And so because they can't bind, those filaments slide back apart. So one thing to note is that when you die, one of the things that happens is you're no longer producing any ATP. And so you still go through this whole cycle. However, you can't break the cross bridge between actin and myosin, because in order to break that cross bridge, you needed ATP. So with no ATP, what happens is that cross bridge gets stuck in whatever position it was in when you died and you stopped making ATP. Now, we call that rigor mortis, and that sets in about 3 hours after you die. One of the reasons it doesn't set in right away is that you can produce a little bit of ATP via glycolysis. Do you remember that? Glycolysis happens in muscle cells, just like all other cells, and it's anaerobic. It doesn't require oxygen to work. And so we can produce a little bit of ATP, and so a little bit of muscle contraction can still happen. But after about 3 hours, it's completely shut down. And so rigor mortis sets in at that time and lasts between 24 to 36 hours after death. At that time, enzymes that are released from like bursting vesicles and bursting lysosomes in the cell are going to break down this connection, and so rigor mortis goes away. So you don't stay stiff forever, you sort of get limp again after that. So to review very quickly, the muscle contraction happens when the signal comes down the neuron and that neuron releases acetylcholine into the synapse, which is the space between the neuron and the muscle fiber. That acetylcholine will bind to receptors on the end plate here, and it will open special gates that are going to allow sodium ions into the sarcoplasm. That will start off a series of reactions that will allow an action potential to run down the surface of the cell, dive down into the T-tubule, and open gates that will release calcium into the sarcoplasm. Calcium will bind to troponin, which will change shape and pull tropomyosin out of the way. Myosin will reach up, bind onto the myosin binding sites on actin, and pull. That's called the power stroke. Then myosin has to bind to ATP to release actin. It hydrolyzes ATP into ADP and releases energy, and it uses that energy to recock the head. If available, myosin will grab onto actin and do another power stroke. This whole thing is called cross-bridge cycling. So we actually need ATP in a number of different places. One place is when we're pumping calcium back into the sarcoplasmic reticulum. Because we're pumping against a gradient, it's active transport, and we need ATP for that. The other place where we need ATP is to break the cross bridge between actin and myosin. No ATP, you're not going to be able to break the cross bridge, and your muscles will be rigid and stuck in whatever position they're in. Where do we get energy for skeletal muscle contraction? Well, some of it is actually stored. So you have about 6 seconds worth of ATP in your muscle fibers. However, at that point, we need to create or synthesize ATP in a couple of different ways. The first way we're going to do it is shown here, and this is where we use something called creatine phosphate, and we take the phosphate off of creatine phosphate and we give it to ADP. So all we're doing is we're sort of exchanging phosphates, and that can create a little bit extra ATP, about 5 seconds worth if you're working out hard. The second way we can create ATP is via fermentation. And remember, this happens in glycolysis. You don't need oxygen for this. It can just happen by splitting glucose into 2 molecules of pyruvate. Recall that in Chapter 4, we studied cellular respiration, and glycolysis was the first step of cellular respiration. You can make some ATP this way, but it only lasts a little while in the cell. The third way to synthesize more ATP is by aerobic respiration. We've already talked about this, and we called it cellular respiration. Now, once you have enough oxygen circulating in your body, you can do this for hours. So that's basically where we take glycogen or fatty acids, we break them down, we send them to the electron transport chain, and we create almost unlimited amounts of ATP. As long as you have something to burn, glycogen or fatty acids, you can continue to make ATP for a while. But remember, this form of creating ATP requires oxygen. That's it for today. See you in class.